Hello, my name is Zachary McLean, and today we're going to be talking about point mutations. So a point mutation is where one base pair is changed in a DNA sequence. And so when you're coding for amino acids, when you're building proteins, there is a three base pair codon that each one of those specifically codes for a different amino acid. So for this, you have ATG to ATC. So AUG as mRNA, and AUC could code for two completely different things. So we're going to go over an example here and some different types of point mutations. So up top here, we have our coding strand of DNA. You can see that highlighted in blue. And then in transcription, you have mRNA that's converted from that DNA strand, and then the corresponding amino acids that are coded for by those three base pair codons. So first we have a missense mutation, and so this is where you code for a completely different amino acid. So it's like in a recipe, when you use baking soda instead of baking powder, you get something completely different. So here we're going from a T to a C. And so you can see it codes for, instead of TRP, it codes for CYS. So next we have a nonsense mutation, which is basically where you go from an amino acid to a stop codon. And so it's like changing a comma to a period in a sentence. That's how you can think of it. So we're going from a C to an A. So instead of TGC, we would have had TGA, and then you get UGA, which codes for a stop. So next we have a silent mutation, and so a silent mutation is like basically where you have a point mutation, but those code for the same amino acid. So it's like having a nail and a screw. They both do the same thing, like if you're joining two boards, but they're technically different things. So from here, we went from a T to a C, and so they both code for ILE. And so the next thing is a read-through mutation. And with a read-through mutation, it's basically where you change from a three base pair codon that codes for a stop to something that codes for another amino acid. So it's like in a sentence, changing the punctuation from a period to a comma. And so here we go from an A to a G. And so you go from ILE to ILE. And so basically the amino acid would keep coding instead of just stopping where it's supposed to stop. And so here are my citations for this presentation. Thanks.